Here we're going to talk about the utilization of fats for ATP production, specifically focusing on the metabolic pathway known as beta oxidation here within the mitochondria. Now, fat use can occur in many cell types for ATP production. That use can go up dramatically in certain circumstances. That occurs especially during fasting, prolonged fasting periods when fats are mobilized as an energy source. Also occurs during prolonged exercise when, again, the carbohydrate stores, which are much smaller, can become limiting. And so what happens is fats are mobilized enzymatically from storage sites in adipose tissue, the liver, and in skeletal muscle. There are actually fat stores within the cells in what are called lipid inclusions. And so in either case, we enzymatically release fatty acids from the storage molecule, which is a triglyceride, to release these free fatty acids. The free fatty acids are then taken up within the mitochondria and processed through this process of beta oxidation. And what beta oxidation does very simply is take a reasonably long chain of carbons within a fatty acid and it cleaves off a two-carbon group forming acetyl-CoA. And then what happens is that now two-carbon shorter chain of carbons can come back, this shorter fatty acid can come back and go through the cycle again, releasing another acetyl group. In many cases, releasing 10 or more acetyl groups per fatty acid. So quite a few carbon groups coming out of that fatty acid, each of which can then enter the Krebs cycle to have its hydrogen atoms released on the carriers NADH and FADH2, which of course can then enter the electron transport chain for ATP production. As you can see, beta oxidation itself is releasing some hydrogen atoms, which can also enter the electron transport chain to help produce ATP. So this helps make fat a very effective storage molecule for energy because we get lots of carbon molecules running lots of cycles of the Krebs cycle. And in addition, it's stored in the absence of water. It's a fat, so it's not a hydrated solute, which makes it very efficient weight-wise for storing calories. And the last thing we want to point out here is that the Krebs cycle and the electron transport chain are directly and indirectly dependent upon oxygen. The electron transport chain directly, because it's the final electron acceptor there. But the Krebs cycle will also not run in the absence of oxygen, making the utilization of fatty acids for ATP production highly oxygen dependent, right? Only aerobic ATP production allowed here.